So next point is our presentations. Today's talk is by Jean-Luc Legras from the French National Institute for Agriculture, Food and Environment, who will be talking about the microbial resources research infrastructure called MIRRI, which is an ERIC, so a European Research Infrastructure Consortium. But first, I will leave the floor to Alice again for a brief introduction.
Yeah, once a month you have me talking all the time. So yeah, I'm really happy to present Jean-Luc. So Jean-Luc, I think, will be present today with several members of MIRRI, as he mentioned. Is here on behalf of MIRRI. He's a research engineer in Montpellier and head of the Levure yeast collection. He's interested in yeast diversity and physiology, and is the leader since 2020 of the work package 2 called Research Strategy of a Horizon project is MIRRI 21. Where it seems that of course, it will explain more during the presentation, but they are also aiming to generate reference genomes. And so, yeah, I think that's it for the short presentation. Jean-Luc, I guess if I missed something, you will add more information.
Thank you, Alice, and thank you, Claudio. I think it's thank you both for, first of all, thank you for inviting me, and I think you almost present everything. So I try to share my screen. Here it is, and I put it in presentation mode. And where it is here. So, is that okay? Do you?
Yeah, now it is.
Okay. So thank you again for giving me this possibility, sorry, to speak about MIRRI and our project. So I wanted to speak a little bit before about MIRRI. So the idea of building this infrastructure last something like 15 years ago, and you see it's quite a long project. It has been, and in 2010 the idea of an infrastructure, a gathering culture collection, has been written in the European Strategy Forum on Just Infrastructure Roadmap, and there is a first phase to prepare this infrastructure from 2012 2016. And if you remember, 2016, there is a major issue with the Brexit and the leave of Kaby, and as well at the same time, DSMZ, which was the pilot of the project, has left. So we had to, the second phase in 2016 to 2019 to reconstruct, reorganize the project. And in 2019, with a new coordination, a new proposal has been made, and they could obtain funding for a new infra dev call, and this project was called MIRRI 21, and the aim of this project was to implement the MIRRI ERIC. And this MIRRI ERIC was actually, I told you, yes, mentioned here, and founded in the 16th of June last year. And we are still building it. So what is MIRRI? MIRRI can be seen as a pan-European distributed resource infrastructure for the preservation, the systematic investigation, the provision, and the valorization of microbial resource and biodiversity. MIRRI ERIC serves MIRRI serves of course, bioscience and bioindustry, but organizing the access into a single point, because you have many culture collections, we could coordinate all of them. And this gave a wide catalog of microorganisms, and also their derivatives, and also data, services, and the domains of course, health and food, agriculture and food, environment, and of course, biodiversity. So using this serving the users by collaborating with other research infrastructure, we're working with the public and the policymakers. So MIRRI ERIC will contribute to the advancement of research and innovation in the bioeconomy and sorry, just what is it? Here it is. So and it will also, we hope that it will be serve as for a sustainable, competitive, and resilient bioeconomy. So the as I told you before, MIRRI ERIC has been set up by the Commission implementing decision last year on June 16, and is the landmark of the European Strategy Forum on Resource Infrastructures, a roadmap. What is it? Sorry. I just cannot. Okay, so MIRRI today gathers about 50 microbial domain biological resource centers. And these are culture collections, also research institute for 10 countries. 5 countries are the founding members: Belgium, France, Latvia, Portugal, and Spain. And Portugal and Spain are the co-host of the ERIC, and the headquarters are located in Braga in Portugal. And your 5 countries are also prospective members. These are Greece, Italy, the Netherlands, Poland, and Romania. So MIRRI has proposed a strategic agenda, and corresponding to these 3 domains: health and food, agro-food, environment and energy. Of course, if you speak about health and food, that one of the field is, of course, the microorganisms connected for research on pathogenic microorganisms, for human and animal infectious disease, but also, and so they can be also microorganisms for the development of new biopharmaceutical or therapeutic solutions, vaccines, fat therapies, or microbiome therapeutics. And if for the agro and food domain, the research are connected for the development of new, safe, healthy, and sustainable food and feed products. And for the more agricultural field, of course, resources for and methods for the biological management of soils and crops. So if you look to the environment and energy field, MIRRI has also resources and methods for biomonitoring, for the bioremediation, for the degradation of organic pollutants, and one field also the research and development for renewable bio-based chemical materials. And of course, last but not least, the last strategic area is about rescuing and preserving microbial diversity. So you can see that there are different domain covers, a broad field of activities. And if you want more, you can go to the website mirri.org that was mentioned in the summary. So who are we? So as mentioned, as already Alice mentioned, that we are a working group of the Work Package 2 of the MIRRI 21 project that was dedicated to research strategy. In fact, our objective is to prepare culture collections to the challenges of new technologies and meet also the future needs of users and of the scientific community. And what we saw is that genome sequencing becomes more and more frequently used, especially for bacteria, but very likely too for fungi, and may become required information for microbial resources. And if you look to the NCBI, to the data we can get, about 62% of known prokaryote species are sequenced, only 2% of fungal species are sequenced. I noticed we counted about 3,000 and fungal species for which a genome is available out of 150,000, and so which means very little. And you could see also this big trend here and in the last decade. So if we look to the data that we can extract from NCBI, we can see that the quality of these genome sequences is extremely variable. And so you can see the high number of genome available for prokaryotes, and you'll see the numbers of figures are much lower for fungi. And here I just chosen to only show fungi. And you can see also is that the vast majority are scaffolds, and we have very few high quality genome at the chromosome or complete genome level. So which means that if we look to prokaryotes, we have something like about 8% of high quality genomes, and for eukaryotes, it was about 17 altogether. But if you look more to detail some taxons, you could have a look for it has here for Ascomycota. You can see that we have, of course, even if you have quite many genomes that have been sequenced from different species, you can see in blue here area in which no genomes are sequenced. And if you want higher level of genomes, you can see the number of genomes is lower. And if you look to complete genome, these are really scarce. This is true for Ascomycota. It's going to be true as well. It's even worse for Basidiomycota. And I could show you the same thing for other also class of fungi. So are there other existing sequencing projects? I give you the example of the 1,000 fungal genomes and 1,000 plus year genomes. These are, and also the WDCM genome sequencing project. These are the three major projects. The two first projects that gave already a very, very nice result, of course, are the two 1,000 plus genomes. And this gives here has brought a new perspective and a taxonomy of fungi. And for the data for the WDCM, however, the progress done on the sequencing are much smaller. But all we can notice that all were done with Illumina sequencing technology. So why should we sequence MIRRI resources? In fact, the first priority was to propose super characterized microbial resources, and we thought that we needed it for that taxonomy of the taxonomic characterization of bacteria, so the bacteria, yeast, fungi, algae as well, and to better explore and preserve biodiversity. It can be a very powerful tool for health, of course, for antibiotic resistance detection, or to better identify fungi that are involved in new outbreaks, like, for instance, the recent Candida auris. It could be also a better tool for better characterization of and exploration of metabolic enzymatic pathways, and also for biotechnology and bioindustry. And as for instance, for biofuel, for wood degradation, and there is a big community of people working on such topic, looking for searching for information on all these all these fungi, and are ready to invest into this domain. And also it could be used for the better characterization of aerosol use for ectomycorrhiza, for fungal pathogens as well. And there's also a big community on fungal pathogens. And also it will also help for synthetic biology. So one major focus which would be, what do microbes do? And for that we need high quality genomes. And to look which microbial which strain should we focus on? We talked about type strains because they're representative of the species. But one difficulty of is that if you look here, that you have without one of the 60,000 entries in the MIRRI catalog, but you have 40,000 bacteria and also 100,000 fungal strains, and altogether about 9,000 type strains. But in fact, we could notice that the type strains are the same and are shared between different collections. So we had to deduplicate these type strains. So in fact, if we look to the MIRRI website, so if we look to eukaryote, you can see that we have only 240 genomes, 40 fungi species with genomes, which are deduplicated, and something like 500 species and 977 fungal species without genomes. In fact, and also for some of the species, the deposit are ancient, so we could not find which was the original strain in the collections. So, but anyway, we can see that for type strains, the percentage of high quality genome is very much lower than even before. So what about the sequencing of facilities and tools? In fact, there are some sequencing platform in the Westerdijk Institute in the Netherlands. University Pasteur also has its own sequencing platform, which is also shared with all the research and labs of the Pasteur. And it's the same for other, in INRAE, we also have a sequencing platform, but are not in-house connected to the culture collection. These are owned by INRAE and shared with other research units. And so in Portugal and Italy, I mentioned them because right now they had a chance to get money for the development of their Portuguese and Italian node, and they plan to invest in different technology, especially for like Illumina technology, or very likely also for nanopore in Italy. And so what about bioinformatics expertise? There is some in MIRRI, but there is of course a lack of bioinformatics expertise as well. So we have some partners in this working group from the University of Torino, from BCCM, from UC Pasteur, from INRAE, but actually that we will also need more, let's say, arms and brains. So what are objectives? We decided to sequence type strands, or maybe also interesting strands in case that the type strands are quite atypical, in order to provide reference genomes. This is why we thought about using long read sequencing, but PacBio or nanopore technology, and maybe in combination with Illumina. And of course, we need to annotate these genomes and also make these data available and visible, and using also try to make it fair as well. So our plans, we want to prepare an opinion paper promoting the genome characterization of microbial resources, bacteria and eukaryotes altogether, because we think there's a kind of lack of knowledge about the microbial resources in cultural collection. And we would like also to apply funding, because we need this money to fuel our project. And so this means we'll include sequencing project in the forthcoming applications. And I think we need to coordinate with ERGA for eukaryotes, for microkaryotes, so we should say. So what do we have? We have well taxonomic characterized resources, especially with type strains. We know the APA and often the metadata of these strains, and we also know most of the time some of microbial strains, and in most cases we know how to extract DNA. We have already some expertise in bioinformatics tools. And what we need, we also need additional expertise, especially in bioinformatics. We need additional bioinformatic tools and training. It has been identified as a key point for the future years inside the MIRRI community. And of course, as I mentioned before, we need funding. And so you think, wow, we thought it would be nice to coordinate with ERGA. So all together, so I would like to thank you for your attention. I will try to answer your question.